So now let us get started with the steps in urine formation. So there are mainly 3 steps that are followed in urine formation. We will have a look into them one by one. So the first step that we are going to discuss about is what we call to be ultrafiltration. So what is ultrafiltration? Let us understand that. If we recall what we have just studied in the previous slide, that in the Bowman's capsule, we have 2 arterioles that are present in this region. What is it? One arteriole is the afferent arteriole, which brings in blood, and the other one is the efferent arteriole, which takes away blood. So what does the afferent arteriole do? It brings in oxygenated blood. Then it comes to the glomerulus. So what is the glomerulus? Glomerulus is a knot of capillaries, we can say. So what happens in the glomerulus is a high hydrostatic pressure develops in the glomerulus. Why? Due to the diameter difference between the afferent and the efferent arteriole. The afferent arteriole is much wider as compared to the efferent arteriole. So due to the difference in diameter between this afferent and the efferent arteriole, a very high hydrostatic pressure develops in the glomerular region. And as a result of that, only the liquid contents of the blood will be filtered out from the glomerulus into the Bowman's capsule. Only the liquid contents of the blood. So this process is known as ultrafiltration, and the fluid which comes into the Bowman's capsule is now known as the glomerular filtrate. So the filtrate which now comes into the Bowman's capsule is known as the glomerular filtrate. Okay, so now let us understand the components or the composition of the glomerular filtrate. So as we just saw that the glomerular filtrate is the liquid part of the blood. What do we know about blood? That blood has distinctly 2 different parts. One is what we call to be, say blood. If we say about its composition, one is what we have the plasma, which is the liquid part of the blood, and the other part we can call to be the cellular part of the blood, which has namely the RBCs, then they will have the WBCs, and then they have the platelets. So the glomerular filtrate is mainly concerned with this, the plasma, the liquid part of the blood. So the glomerular filtrate has the plasma along with the organic and inorganic substances like glucose, amino acids, urea, etc. So majorly the components of the glomerular filtrate are water, urea, salts, glucose, and various other solutes that have been dissolved in the plasma. That is about the first process, which is ultrafiltration. Now coming to the second process that we have, what we call to be selective reabsorption. So if we analyze this term a little carefully, selective reabsorption, it gives us quite some of its meaning. One is selective, that the tubule knows what thing to reabsorb. It does not reabsorb anything and everything. It will only reabsorb those things which are useful for the body. Right? So the glomerular filtrate, which has entered into the proximal convoluted tubule from the Bowman's capsule, that is a very dilute kind of solution because it only has the liquid part of the blood. Along with that, what we have just seen is it also contains usable substances like salts, glucose. So our body does not want to waste all of this by passing it out through the urine. So what the body does is the proximal convoluted tubule reabsorbs back much of the water that is present in this glomerular filtrate, and along with that some amount of glucose, sodium, that is also reabsorbed back. So basically water along with the usable substances are reabsorbed to a great extent. What we have to keep in mind here is that this reabsorption that takes place, that occurs in such a manner that the normal concentration of the blood is not disturbed. It is taken into consideration that the reabsorption needs to happen in such a way that the general concentration of the blood flowing in the body is not disrupted. So this is what we call to be the selective reabsorption. So some part of selective reabsorption occurs in the proximal convoluted tubule. Then in the loop of Henle also, what will happen is in the descending part of the loop of Henle, water is being reabsorbed. In the ascending part of the loop of Henle, the NaCl, the sodium chloride, the salt is being reabsorbed. And then further into the distal convoluted tubule, again some reabsorption also takes place. So this is about selective reabsorption. The third process that occurs in case of urine formation is what we call to be the tubular secretion. So what is this tubular secretion? The name itself again gives us the meaning. That is, the tube, the renal tubule is secreting something. What is it secreting? Say there are certain substances like K plus ions, potassium ions, or foreign chemicals like penicillin or other drugs. So they are passed into this glomerular filtrate, which process is what we call to be tubular secretion. Okay, so what came after ultrafiltration is glomerular filtrate. That underwent the process of selective reabsorption, wherein what happened was water, glucose, sodium, so water with the usable substances got reabsorbed back. And then in the DCT, we saw the process of tubular secretion. That is, penicillin, potassium ions, etc. got secreted into this fluid. And ultimately, what we get, that is the urine. Okay, so this is a very quick recap of what we have just studied. That is the function of the different parts of the renal tubule. So the first one that we have here is the glomerulus. So as we have seen, glomerulus has the role of ultrafiltration. And what is ultrafiltration? That the liquid part of the blood will get filtered under the high hydrostatic pressure from the glomerulus into the Bowman's capsule. So the role of the Bowman's capsule is in receiving this filtrate, the liquid part of the blood, that we call to be the glomerular filtrate. Then we have the proximal convoluted tubule, the next part, which is abbreviated to be PCT. So it has the role of selective reabsorption. That is, it is going to take back most of the water and usable substances like glucose and the salts, that is Na plus Cl minus ion. Then we have the U-shaped part, which is the loop of Henle, that continues the role of the selective reabsorption of water and sodium ions. Then we have the distal convoluted tubule, which continues some part of the selective reabsorption, and the tubular secretion function, which is basically secretion of substances like penicillin, then K plus ions into the glomerular filtrate, and ultimately the urine goes into the last part of the nephron, which is the collecting duct. So this basically shows the entire picture at once. That is, the blood comes into the renal artery. Then the renal artery has its branches, out of which one is the afferent arteriole. So the afferent arteriole will enter into the glomerulus, wherein ultrafiltration occurs. Once ultrafiltration occurs, the liquid part of the blood will get into the Bowman's capsule. That is what we call to be the glomerular filtrate. And the thicker part of the blood will flow out via the efferent arteriole. So we can say that the efferent arteriole contains the thicker part of the blood, because the liquid part has come into the glomerular filtrate. Glomerular filtrate then gets into the Bowman's capsule, and from the Bowman's capsule it will go to the PCT, where we see the process of selective reabsorption to be beginning. From there it passes to the loop of Henle. The loop of Henle will also continue the process of selective absorption. From there it will go to the DCT. From the descent, it ultimately goes to the collecting duct. So at this stage, where the fluid goes from the descent to the collecting duct, it is now known as the urine. The urine then goes into the pelvis, which is a part of the kidney. From there to the ureter, to the urinary bladder, to the urethra, and finally outside of the body. Then, so from the collecting duct onwards, this is the pathway to the pelvis, to the ureter, urinary bladder, urethra, and outside. One thing that we are noting here is a subtle difference between the male body and the female body is that in the female body, the urine directly flows from the urethra to outside of the body, while in the male body, we'll see that there is the penis which is involved. One more thing to be noted here is the movement of urine in the ureter occurs by a special process that we call to be peristalsis. What is peristalsis? Peristalsis refers to the fact that there is an alternating movement of contraction and relaxation of the ureter. So the urine gradually passes from the kidney to the urinary bladder, right? And ultimately, via the nervous influence on the urinary sphincter muscles, this process of micturition, which we in simple words call to be urination, that is the passage of urine from the body to outside, occurs.
So that is all about the topic of urine formation. Please like the video, drop a comment and share with whoever you think might benefit from the video. Also subscribe to our channel Bioexplaining. Thank you.
See, the left hand side figure shows a very distinct calyx and corolla. This is what we call to be the stamen of a flower, the male part.